Hello, everyone. Welcome to Lunch Break Science. I'm Ariel Johnson, your host from the Leakey Foundation, a nonprofit dedicated to supporting human evolution research and sharing discoveries. I am thrilled that we're back live with a new episode. Today, we look at how we evolved our particular type of locomotion in the field in Kenya working, and so much more. Before we jump in, we'd like to thank the Ann and Gordon Getty Foundation, Camilla and George Smith, and the Joan and Arnold Travis Education Fund who made this episode possible. Now, let's bring on our guest, Leakey Foundation grantee and paleoanthropologist, Dr. Carol Ward. Carol, thank you so much for being here with us.
Hi, Ariel. Thank you for inviting me. I'm really thrilled to be here. As you know, the Leakey Foundation started me off on my career many, many years ago. And so it's really fun to be still involved to be back today. So thanks.
Well, we're excited to have you. Carol is joining us from Missouri, where she is a curator's professor of pathology and anatomical sciences at the University of Missouri. Carol co-founded the West Turkana Paleo Project alongside with past lunch break science guest and Leakey Foundation Baldwin Fellow, Dr. Frederick Chalamanthi, and Dr. J. Michael Plotkin. Carol also is on the Leakey Foundation Science Executive Committee and was on the very first episode of our podcast, Origin Stories. Before we hear from Carol, if you are watching this episode live, please post comments or questions in the chat, and Carol will answer those questions live during the episode. The earlier you get those questions in, the more likely your question will be featured. Carol, we humans are found everywhere. Why is it important to study the evolution of our locomotion and how we move around the world?
Well, humans are an amazing species that live all over the world and have language and culture and technology and all the amazing things. But the story of human evolution begins at the beginning. So if we can understand something about our earliest ancestors, we know the basis that selection had to work on that shaped the evolution of our genus Homo and all the amazing developments we've made after that. So the thing about our origins, one of the first things that set us apart from apes is how we move around the world, like you see in that next slide, I think. If you compare us to every other living ape, they're all pretty similar. They hang in trees, they use their arms, big long arms to move around in the trees, and we don't do that. And comparing apes to humans gives us the basis for that Leakey Foundation logo, like you can see here. Which was published in Time Life 50 years ago. And it depicts us evolving from a modern ape-like ancestor and turning into humans. But that may or may not be the case. Let's go to the next picture. And we have this idea that if you evolve from an ape into a human, the big question of our origins that set this amazing trajectory of human evolution on its course was, why did we stand up from all fours like the chimpanzee uses? But now, since the last 50 years, we have thousands and thousands more fossils. There were probably hundreds of fossil apes that lived in the past. And if we study all of these, we're beginning to get a different picture. So go to the next one here. A lot of these apes weren't quite like modern ones. And they make us wonder, maybe the question we should be answering is not, why did we stand up on all fours, but why did we never drop down on all fours to begin with? And it's the fossil record that lets us address these kinds of questions and understand what happened in our origins. And therefore, we can begin to understand why. And once we get the origin of hominids correct, we can then get a much better picture of how everything progressed after that. So that's why it's important.
I'm just really excited to get to your talk, but before that, we have some questions. I want to know more about you. What inspired you to focus on this area of research?
Well, if you listen to the Origin Stories podcast, I tell the story in more detail. But I was at University of Michigan and I wanted to take a weird class. So I took biological anthropology. Our friends told me it was cavemen and stuff. I thought that sounded cool. And I took it and I fell in love with it. To see casts of animals that look so different from us, but are our fossil relatives, was just fascinating to me. And understanding how we can use those and study those to understand how evolution took place is what's occupied me most of the time. in my career. And in the last 15 years or so, I realized that studying fossils is great, but I wanted to get out to the field and I wanted to find the fossils, which is why I teamed up with Dr. Monthi and Dr. Plofkin, and we've been working in the west side of Lake Turkana ever since, with this West student here, Sharon Kuo, finding new fossils that reflect our ancestors, their environments, and what the world would have been like when they lived. So it's really been a lot of fun.
It sounds that what is the importance of mentoring others and inspiring them?
Well, we scientists can do all the science we want, but if we don't reach out and share what we're doing and help the communities we work in, we're really not doing our job. So one of the key keystones of our field project is that we work with local communities where we find our fossils and we ask them what they need. We've helped build 2 primary schools and 2 of our field sites and try to help kids get education so someday they perhaps can pursue the study of human evolution as well. But we don't just do outreach in Kenya. We also do lots of outreach here in the United States to help make a more scientifically literate public. Getting kids looking at fossils is a great gateway science to studying science in general, and people love it. And so we feel very strongly about bringing what we do to the public, even here in the US. And in some ways, one of the most important things we do as paleoanthropologists and professors is help educate the next generation, mentor students at the graduate level, at the undergraduate level. These are my students now in my lab. We are partners in everything we do, and we have undergraduates, and we reach out and train them, and they go on to positions. Here I am with Dr. Don Johansson and Dr. Ashley Hammond, one of my students, who's now a curator at the American Museum of Natural History. So we're giving forward, sharing what we've learned so the next generation of younger, brighter, more energetic people can take over.
Well, now I can see your influence because when she was on Lunch Break Science, she was just an amazing and wonderful presenter. So I think she probably picked up some of your skills. Uh,
Well, I'd love to claim credit, but
Carol, you're a member of the Leakey Foundation's Science Executive Committee. Tell us about this role.
this is one of the things I think most passionate about my career, because again, this is a way I can not only see what's going on in the field firsthand as we see the proposals come through. I get to work with some of the brightest, most influential scientists in our field, and we get to help give people that start and help pursue science as careers and help more established investigators do the research. I think probably everybody in paleoanthropology, or at least many people, have been either directly funded by Leakey Foundation or influenced by Leakey Foundation awards over the past. And it's so fun to be able to be part of that process. It's just great.
It is. And, luckily, we were able to award almost twice as many Baldwin Fellowships as we've received in the past. We actually have a clip of our current cohort of Baldwins. The Leakey Foundation's prestigious Baldwin Fellowship program supports advanced education for a global community of scientists. As I mentioned, the 22 Baldwin Fellows you're seeing now, it's I think 2022 as in the year 2022. We were able to offer, as I said, twice as many fellowships as we were able to offer in an average year, which is just absolutely wonderful. We shared a link in the chat for you to learn more about this year's fellows and their works. The Baldwin Fellowship Program is nearest and dearest to the heart of our executive director, Cheryl Camisa Smith, and today is her birthday. Happy birthday, Cheryl. She has made it a goal to raise $1,000 for the Baldwin program as part of a larger effort to continue to expand the successful program. You can join in the celebration and help support the next generation of leading scientists. We've shared a link in the chat to learn more.
I'd just like to pop in and say how impressed I am with the Baldwin Fellows. One thing that we really need to do is get international scholars involved in research. So it's not just Americans, Europeans doing most of the work. And it's really great to see these brilliant young scientists who are able to have a chance to contribute to our field in an intellectual capacity. And it's just terrific. Working with Chalo, who was a Baldwin Fellow all those years, working with other of my colleagues who are Baldwin Fellows and got their start thanks to this program. It really couldn't be more important. And I think one of the most important things the Leakey Foundation is doing these days.
Absolutely. And we funded Baldwin Fellows from Azerbaijan to Zimbabwe, so and everything in between. And we've shared a link in the chat to learn more about helping support the Baldwin Fellowship program. Carol, you are one of the co-founders of the West Turkana Paleo Project in Kenya. What is it like working in the field in the Turkana Basin?
Well, it's hot. It's really exciting to be able to go to somewhere that very few people outside the country, a place they don't get to go see and experience and find evidence of animals that lived millions of years ago that have never been seen before is really terrific. But it's really challenging work. And it's very hard to get out there. The roads are distant. It's long drives. We get big camps of a big lorry. We have several vehicles full of equipment, which inevitably break down somewhere. We have terrific mechanics that can fix anything because, as you can see, the roads are maybe not quite as good as some of the roads that we drive on here in the US. So it's quite an effort to get out in the field, but absolutely worth it. And as we travel these roads, things become more and more and more rural until we get out to these very desert environments, which is where we can see the fossils. This is where they've been exposed. This is where there aren't cities and jungles and things on top of them. And we can actually find them. And we have a large team of people, maybe 25 or 30 people in our camp. I work with some of the most accomplished and famous fossil finders in the world. And we're a team that works together. We bring students to the field from various places. We have scholars. We work as a team and we live as a team. We have a wonderful camp. And people make camp very important, bring important things like showers at the end of the day, which let me tell you is about the best thing. We are very comfortable. Here's Sharon again, enjoying a hammock as we're avoiding the heat of the day, midday. But most of the time we spend out walking and looking at fossils. All these rocks you see might be fossils. We work again closely with the local community. Here's another former student, Elizabeth Moffett, who's also a Leakey grantee, meeting some of the local people around Canapoi, again, to work with the community. So we get to know the people there, and they've been so gracious and wonderful letting us come into their home and their place that they live and do our work. And we have, like I say, a large camp, and we have cooks who can cook anything. We have an office. We have some solar power to charge our equipment, a fridge. We have water filters. We have comfortable, shady tents. We have people who help us keep the camp safe and clean and organized, which we couldn't do without. And importantly, we act as one camp. We all eat together as a team. We live together side by side. Here are cooks making mandazi, which are a fried bread kind of a food for our breakfast. So we have delicious hot meals because it's a lot of hard work out there. And they work very hard to keep us going. We all eat family style. We eat all Kenyan food around a table, which when we're lucky, we get greens, a lot of potatoes. And these goats aren't just pets, I'm sorry to say. But they make it very comfortable and easy for us to do the science, and we're all in it together. So it's really great. It's a family.
Oh, it sounds wonderful. Your lecture will start pretty shortly, but I have one last question. What is your favorite fossil?
Oh, it's so hard to decide. There are so many great ones that I've worked on over the years. Some aren't very impressive. But I think the most impressive, in some ways enigmatic fossil might be a fossil that was called the black skull because the color of the bone is very dark. It's about 2.5 million years old. It's from a site called Lumaquee, where I am now working. It was found in the 80s by Alan Walker and Richard Leakey. And this is one of the robust Australopithecines. You can see the big crest on its head. It has this giant face, huge chewing muscles, but a lot of primitive features. And it has so much to say about the branching pattern and how all the lineages diverged. We need to know more about it. We have a few more fossils from Lumaquee that I hope will help us. But I think that's probably the coolest fossil.
Oh, it is a beautiful one.
It is gorgeous.
So before we turn the virtual floor over to Carol, if you are enjoying this episode and want to learn more about human evolution, amazing research going on all over the world, and the latest discoveries, start by subscribing to our channel and clicking the notification bell so you don't miss a thing. Now, let's turn the virtual floor over to Carol and hear more about her research.
I'm going to have to go back to the beginning here. There we go. All right. So what I'm going to talk about today is a new project that was actually just funded this cycle by the Leakey Foundation. And I put it in with my colleagues here at Missouri, Dr. Casey Holliday and Kevin Middleton. Casey and Kevin are not yet paleoanthropologists. Casey is a biologist, mostly studies archosaurs and feeding behavior and how heads works and dinosaurs and crocodiles and other things. Kevin is an expert in biomechanics in a variety of different ways. And in talking with them, we've come up with a project that pulls our expertise together to say, I hope we'll say something new and really exciting and informative about the anatomy and biomechanics of the hand and feet in chimps, gorillas, and humans. Also on this picture here, you see my lab members. Eden Makareth and Mara Fields did a lot of the work on the pilot project I'm going to talk about today. Austin Lawrence is working on pelvises and kinematics funded by the Leakey Foundation also, but he's helping us out along the way. So everything I'm talking about is a team effort, has been, and certainly will be moving forward. So we talked a little bit about my work in the field and going to find fossils. It's hard, it's expensive, it takes a lot of knowledge on part of a lot of people to do. But equally difficult is when you get all these little fossil bits and pieces back to the lab, is to organize them and then try to figure out what they tell us about the animals lived in the past. What were these creatures like, and what does that tell us about our evolution? And in some ways, that's a really hard part. For example, I talked about locomotion and the importance of walking upright on 2 feet. And that involves, of course, the foot, because that's what's on the ground. If you see in the upper left here, there's a chimp foot compared to a human footprint. We are the only primates that don't have a grasping big toe that is really useful for moving around in the trees. Instead, our toes are all lined up with the rest of the digits. Apes can use these feet to grab around the branches. They're flexible. It's really useful. And we gave that up. You could see footprints here in the center. These are 3.5 million years old, or a little bit more, from Tanzania. And this tells us that some of our earliest ancestors are already walking upright on 2 feet like us. And that's why this transition is so important. But feet are pretty complicated structures. Here's a human and some great ape feet. There's the skeletons, and they all look different. The size and the shapes of the bones are all proportioned differently. And this is related to how animals use their feet. And you can see how complicated this is with so many bones, but actually what determines what a foot can do is the muscles, the ligaments, the tendons, and the soft tissues inside those feet. And we not only don't get whole feet in the fossil record almost ever, we certainly don't get any of the soft tissues. So it's really hard for us to try to figure out what foot bones in the fossil record tell us, especially since often, or most often, we tend to find maybe one at a time. We wrote a whole paper on one bone from the middle part of the foot, and we think it said something important about feet. But what we do is we use the size and the shape and the proportions and the orientations of the joints to make these inferences. But often we're making inferences. A similar problem happens with the hand. Hands are clearly extremely important for humans, notably for manipulating objects, for making and using tools. And this is a huge focus of interest in paleoanthropology. When did we begin making tools? How did it work? And how was our anatomy and our brains built to make all of these tools? The brains we can't talk about, but the hands we can. And again, hands, like feet, are very complicated, and often we find one. Here's another fossil that our project recovered in Kenya. It's almost 1.5 million years ago, and it's a metacarpal, a bone from the middle of the palm, like you can see in my hand here at the bottom of the slide. And on the bottom of that bone in this fossil, which is the brown one on the right, you can see a little point of bone. And that looks like a human, which is the one in the middle, a little point of bone that you don't see the chimpanzee on the left. And from that, we assume that the bottom of the bone here that articulates with all the little wrist bones in the hand is shaped different in a human from other animals because of the forces that that thumb and that hand is subjected to when grasping. This is a hypothesis based on the work of Mary Marsky, done by my colleague Matt Tosheri, that suggests that all the orientations of all those little bones in the hand and the wrist reflects the loads and the forces of the different proportions and muscles in the hand as they're used. But again, without really understanding the soft tissue anatomy, we don't really have a good idea what those forces would be, the directions, the strength, and everything else. And that's where Kevin and Casey's work has come in. It turns out there are new ways to visualize the anatomy of modern animals. If you go to the doctor and get a CT scan when you maybe have a problem, this on the left is pretty much what the resolution looks like. It turns out you can now soak soft tissues in a solution containing iodine and actually visualize the soft tissue. So in this view of the same hand, you can see the muscles, you can see the nerves in the center of the hand, you can see blood vessels and all kinds of things. From that, what we can do is we can do some pretty nifty science. So we can dice CT hands and scan them at very high resolution. We can segment muscles and things into 3D shapes. This is the work from one of our students, Spiro Sullivan, here at Mizzou. Then we can use machine learning to identify every little fascicle or fiber within the muscles. And we can use these to quantify how strong the muscle could act, the orientation of those, and then calculate the biomechanics of the system and how it works. So we're going to try to take this method that Kevin and Casey have developed, apply it to hands, and see how it works. And we've done a little pilot study. We think it's going to work because, again, former Leakey Foundation grantee, Dr. Faye McGeechee, was able to use this to study necks, another really complicated anatomical system, sort of like a hand or a foot. And you can see at the top here, she's able to segment all the muscles. She's able to make 3D shapes with them. In the center, she's been able to identify all the fascicles. And on the right, you can see the orientation of the many, many muscles that are in the neck. And from that, she can look at the biomechanics of how primates use their neck in new ways. So we're going to do this for hands and feet. So here's some pilot work that was done by Eden Makareth and Mara Fields, showing dice CT images of a human and a chimpanzee. And you can see the very fine detail of every muscle fascicle visible in these images. These are the kind of imagery we've never been able to use before, we've never had before. We've never been able to see the anatomy in 3D all at once at one time. In the past, anatomists have dissected and taken apart hands piece by piece, but then they're taken apart. Here we can see everything all at once. And what we do when we look at just say the muscles that run the thumb and the little finger, we can compare different animals. So here's our human compared to a chimpanzee. Each little noodle here is a fascicle of the muscles. The color differences indicate the orientation, more longitudinal or more sideways. The anatomists in the crowd can enjoy all these long muscle names, but even if you've never taken anatomy, you can look at this picture and see the muscles running the thumb and the human here are very, very different from what we see in the chimpanzee. We can even see differences in the muscles running the little finger. And if we put more muscles on here, you would see even more differences. This is really pretty cool. So what we're going to do is we can use these muscles and we can calculate how strong the muscle would have been. If you go to the gym, the people lifting the heaviest weights probably have the biggest muscles because they have more fascicles involved. We can measure that using this method. So for example, these two muscles on the left here are the ones that really pull that thumb across the hand and bring them in opposition to the other digits. They're much, much stronger in a human than they are in a chimpanzee. And we can drill this down into more details and add more muscles, but it gives us an idea of how it works. We can also look at the orientation of the muscle fibers, because if you pull on something up and down, you're going to get a different result than if you pull on something side to side. So the orientation is really important. And these are just 4 of the muscles that run the thumb. The details aren't super important, but we can take each fascicle and plot it in 3D space. And this is basically just a graph showing the orientation in 3 different dimensions. And again, you can just look at, say, Opponens pollicis, this muscle here. It's very big in the human. It's very small in the chimp. And in fact, there's so many dots on the graph for humans, you can't even really see the chimps. But they're fairly similar orientation, but very different in size. On the right-hand side, for example, the 2 muscles on the right-hand side here, you can see chimpanzees have a rather different orientation than you see in the humans. And the different orientation is going to give a different result when those muscles contract. We can also, if you look at the bottom left, this last muscle here, flexor pollicis brevis, has some differences in orientation. The human has a little more sort of up and down, if you will, orientation to the fibers. But also, if you look at the base of the thumb, it's much further away. And how far away the muscle is from the pull, again, affects how strong an action it can effect. So how strong can you grasp? Sort of like you try to open a door, pushing by the doorknob, which is far from the hinges, is easier than if you push on the door next to the hinges. The same thing happens with muscles. So using this method, we can actually get the strength, we can get the orientation, and we can get the position. And from that, we can get the biomechanical capacity of a chimp hand here compared to a human hand. You can do this for more animals, you can add more muscles and get the whole 3D biomechanical environment. So this is what we propose to do to the Leakey Foundation, and we're very proud that they think that it's worth doing. So the method is really cool because, like, we can see everything all at once in 3D. I'm not showing you all of it because it gets confusing. This is non-destructive, so we don't have to dissect apart these animals to look at them. We can do it on the computer and leave our specimens intact. And when you have very rare animals and very rare specimens, this is really important. Someone can come back and look at these specimens intact in the future. And we can calculate all of the biomechanics of the hand and see how they work differently, how the muscles work differently in different animals. And then using a method, again, piloted by Drs. Holliday and Middleton and their former student, Dr. Caleb Sellers, who's now at the University of Chicago, we can look at the orientation of the joint surfaces and map it to the muscle pull. We can look at the size and the shape of the bones and see how does that reflect the muscle pull and how this works. And once we have that, we have bits of bone and information about those that's informed by the soft tissues that we can take to the fossil record, like some of these spectacular hand fossils here, and some of the more individual bones here and there, and we'll have a knowledgeable basis. What do these joints and bones and proportions tell us about the muscles and what the animals can do? So we think it's really exciting. We're going to take these pilot data, take the show on the road, if you will, and compare chimps and gorillas and humans. We're going to look at the interplay between hands and feet. How do they work differently? How are they similar? How are they different among taxa? And what can we see from this in the bones? And then the next step will be to go back to the fossil record and see what we can say about the origins of locomotion, suspension, bipedality, all of the things that apes and humans do. So we're pretty excited about all of this, and we're looking forward to getting going. But before I end here and maybe take some questions, I'd like to thank some of the funding that's gotten us this far along the way, the people that have helped us with specimens and so forth. So I hope that you get an idea from this about we can find fossils, we can measure fossils and compare bones to one another, but the skeletal system is only a small part of what makes us humans or what makes a chimp a chimp. And now with new imaging technologies, we're able to see things and measure things and quantify things in new ways that'll give us the ability to eventually do dynamic modeling of how animals may have worked someday down the line. So as scientists, paleontologists, the fossils are important, but coming back to the modern animals and figuring how they work and understanding how they work gives us tools to take back and understand those fossils. And we're hoping that this project and ones that'll follow it will help us understand human evolution much better, and then maybe be spun out to look at the evolution of lots of other different animals. So with that, I'd just like to say thank you. And if you have any questions about hands or feet or scanning or fossils or field work, I'd be more than happy to take them. Thank you.
Carol, it was a wonderful talk. Thank you so much. We're going to be taking some questions from our viewers. So if you haven't submitted your question, get those questions in the chat right now. I have a quick first question to kick us off, though. You and your team had to shift and adapt for your projects. As a scientist, how do you learn this skill?
Shift and adapt for COVID, you mean?
Oh,
yeah.
Yeah.
Well,
anything.
Sure. Well, I came from an anatomy background as my graduate training at Johns Hopkins with a man named Alan Walker. And Alan always stressed the importance of having informed and grounded ways to look at your fossils. We can make all the stories we want, but that doesn't get us very far. We need to test our ideas. We need to validate our work. So through my whole career, I've kept a lab component and a field component going. And I encourage my students to do the same because they feed back to one another. The fossils get us the questions that need to be answered, and the studies of modern animals can help us answer some of those questions. So it's been a pivot for me all the way along. And also when there are things like COVID, you can spend a little more time in the lab catching up with some of the analysis before you go back to the field.
Our next question comes from Sarah. Sarah asked, I've heard the idea that our ancestors might not have walked on all fours. Can you tell us about that?
Oh, I could do a whole lunch break science on that one. Yeah, it's true. When we look at the apes that lived in the past, there was a period from about 22 to about 5 million years ago or so called the Miocene. And there were dozens and dozens of ape species that lived during that time. And not a one of them looks exactly like any of our modern animals. Their modern apes are some are very large, some are small. They're all very specialized. And as we're learning how these fossil apes fit into the modern apes, what we see is that tree hanging behavior of African apes, of orangutans, of gibbons may have all evolved separately in those different lineages, and perhaps even to some extent between chimps and gorillas. But they have very specialized anatomy to do this. So when chimps and gorillas spend time on the ground, which they do a lot, they tend to drop down on all fours. They've got big, heavy, strong arms. They've got hands that don't even really uncurl and flatten out all the way. So they walk on their knuckles. And this is how they move on the ground. But none of the fossil apes looked quite as specialized or quite as large as African apes. And their body build is a little bit more generalized. So they probably moved in the trees, holding their bodies upright like modern apes. But because they were able to perhaps stand upright better, when they came to the ground, they may not have had to drop down on all fours. And this is an idea that's sort of coming of age, if you will, within the literature. But that's what the data is suggesting, that at least there's a strong possibility that maybe our ancestor wasn't a knuckle walker, which means they may not have been on all fours when they're on the ground.
That was a great question, Sarah. Now let's hear from Jean. Is a human grasp, say in parallel bars, is it compared to a chimp grasp? What shows as human adjustment compared to the same for chimp?
Sure. If I get your question, Jean, let me know. If that's not, let me know that too. But if you look at our hands, we have long thumbs and pretty short, stumpy fingers. Apes have very long fingers and very short thumbs. So when we grab things, we tend to use all of our hand, our thumb and our fingers together. When we hang, sometimes when you go to the gym and do your pull-ups, you're really just using your fingers. Chimps and gorillas and other apes have very long fingers that let grasp long branches, and they can sort of do a hook much better than we can hanging in the trees. But what our long thumb and short fingers lets us do is a nice pad-to-pad grip, which chimps can't do because their thumbs are so short and their fingers are so long. So the way they grip things can be done very differently. If they're going to hold something small like a pen, I can hold it between my thumb and fingers. A chimp kind of has to do something more like this. So the proportions of the hand and the muscles involved differ, and we can grip things, but we just do it differently. And we think that lots of animals do it differently in lots of ways, but we haven't had the tools to study it yet. So with any luck, tune in a year or two and we'll have some even better ideas.
Well, we'll have to have you back to talk about it. Our next question comes from Hamilton. It was an early question, so there we go. Uh, is there a school in Kenya for paleoanthropologists? There are 2 high school students from Marsabit region next to Turkana interested in studying human origins, and they're interested in helping.
As far as I know, and things are changing very quickly in this, at Turkana University College in Ludwar, there is now actually a master's program in hominin origins, and there's actually courses they can take there. At the undergraduate level, I am less familiar with the offerings there, but you can find that out. There are very actually few undergraduate majors in paleoanthropology in Kenya. It's not very popular, but that's a place that's really becoming a center for getting Kenyan students to be able to study. And so you might check into that.
Great advice. Our next question comes from Kay Lindsay. You've done a lot of transdisciplinary work. What are examples of fields that you've worked in order to incorporate new perspectives? What seems to be the most unusual but proved unexpectedly useful?
Well, the answer to that is in some ways this new project, just to start to go backwards to forwards. I've been working in this department with one of the leading experts in cranial functional morphology, study things like T-Rex cartilage and how alligators bite hard and so on and so forth. And I have someone that Dr. Middleton that has done experiments with mice that have been selected for exercise and archosaur feet and all kinds of things that I wouldn't necessarily have thought about being directly relevant to what I've done, but because they've come up with this phenomenally new, cool workflow to study biomechanics of complex systems, I'm actually entering in some ways a new area for me looking at soft tissue anatomy. So that's been really fun. In the past, learning about new ways to do scanning and measuring things in 3D and morphometrics has been really fun. But I think this is my newest and most significant departure, and it came from a field outside of paleoanthropology. So it's really fun. It's fun to kind of do something new and bring something new into what we do.
The amount of collaboration, and it's just got to be so amazing and so eye-opening. We have a question from Pete. So Pete would like to hear your perspective as a musician about variation in human hand size and what you might have to say in regard to that.
Well, certainly there's a lot of variation. If you just go out to walk down a city street and look at people, our proportions are very, very different. But there are things that we all share. No matter whose human hand it is going to be more like another human hand than it is going to be like the hand of a different species. So we're certainly really interested in how species vary within a species, what that means for function and ability. And clearly variation is what selection acts on over time. So those are really important, good questions. And I think the approaches we're taking may be a great way to get at some of this kinds of questions. We can only work with cadaveric materials, so we can't do this with living people, image people, at least yet, though I imagine that will come pretty soon. Their technology's on the horizon. So that's a great question. It's one we're not going to be able to get to yet, but that's the kind of thing that we hope this line of inquiry will lead open to the possibility of discovering in the future.
Our next question comes from Brandon. Can you talk a little more about how this new technology was developed? Was it originally created for another purpose, or was it designed for bioanthropology from the ground up?
It was absolutely not for bioanthropology at all. Casey and Kevin have been working on trying to understand feeding behavior and crocs, dinosaurs, birds, and their relatives, and understand the importance of 3D anatomy, given the differences in the evolutionary histories of those groups. And they're the ones that have really pulled together this workflow. And I've been watching them over the last few years. They have a student, Spiro Sullivan, that did that great hand picture I showed. He's using this to look at the pectoral flight muscles of birds and the evolution of that and the bony structures that support all of those. So their lab has been pioneering these methods for the last number of years in sort of vertebrate functional morphology. And I thought it would be a great opportunity to take that workflow and apply that to questions from paleoanthropology. So it's really a fun collaboration in which we all bring something rather different to the table and then create something that the whole is more than the sum of the parts, I think.
We have, I've got two more questions. Let's see here. Is it? Well, before we get to I have a question. So you talked about your favorite fossil earlier, but is there any particular fossil that's just really perplexing to you, even now still?
Well, that's why that black skull is really perplexing. Why I like it, because that's really perplexing. Certainly, the new fossils that are coming out that are much more recent and very primitive, like Homo floresiensis, the Homo naledi. Homo floresiensis is the hobbit fossil, which is the press called it. It's these very tiny animals from Southeast Asia. There's much more small-bodied Homo naledi fossils, maybe 250,000 years ago in South Africa. They don't make any sense in what we always thought would happen, but that's the best thing about new fossils is they bring you with new questions. So those are really enigmatic in terms of what they say about how all of evolution progressed. And there are so many new fossils being discovered now that it's almost like fruit basket upset on a number of different questions as we're trying to figure out what all these animals mean and put them all together, put all the new information together. So it's a great time to be in paleoanthropology, I gotta say.
Our next question comes from John. John asks, how has COVID impacted your research?
Slowed it down. We were very lucky because with an international collaboration, Dr. Monthi, who's our Kenyan collaborator, was able to actually, with the Kenyan team, operate and continue the field work when some of the Americans couldn't get over there. So our project was able to keep going and collecting fossils. Mostly it just really slowed us down on getting over to the museum, analyzing the fossils we have, a lot of which are still sitting in bags because the scientific team hasn't been able to come in and do all the study. The lab work here was able to pretty much keep going, but it slowed everything down. And it's really fun now to be seeing everybody getting back in the field, getting back in the museums, picking the work up. Our team especially, I was finally back in Kenya for the first time since the beginning of the pandemic, looking at some of the fossils that have been found since I was able to be there. So I think we're getting back at it, but it's been really challenging for everybody.
Okay, there's one last question. I think we have. Pete asks. It's a question for me. How did I get into paleoanthropology? Well, for me, I think I loved old horror films, and actually, you can see in my background right here is a fossil hand, and it's actually from the movie The Creature from the Black Lagoon, and there's this portion where they're talking about evolution. And I was really fascinated by change and just all of these concepts, and I wanted to learn more. And when I was in college, I was originally a biology major, and I got bit by the anthropology bug by accident because the biology department was impacted. So anyway, I took my first class on fossil humans and was just, never looked back. So yeah. That is how I got into paleoanthropology.
There you go. Maybe we could scan the Creature from the Black Lagoon's hand.
Ooh, I like that. People ask me if it's a whale, but it's kind of close. Yeah. My last question for you, Carol, is: do you have any advice for those just starting out or hoping to pursue a career in science?
Absolutely. Get involved, do science. The best way to learn about what science is like and how to do it is to do it. If you have a museum or a university or a college or anybody doing anything with the kind of science you're interested in, get involved, show up. Scientists are almost always will love to have people come in and get involved and help out. Also, read everything you can, attend talks, watch Leakey Foundation podcasts and some of the great other web resources that are out there. Get involved. Reach out to people doing the work. Don't ever feel intimidated, because most of us love to have interest. We're professors. We love to talk about what we do, but we love to have interested people come in and share our excitement for what we do. So the biggest thing is get involved, listen, read and listen to things outside your field, like I was able to do for this research project. Just get your feelers out there, read, learn, study, and talk to people. And that's the best thing you can do.
Well, that is some amazing advice. Thank you again so much for joining us, Carol. It was a wonderful episode.
It was so fun to be here. I could sit here all afternoon. This was great. And thanks to everybody who sent in great questions. And happy birthday, Cheryl. And thank you again for having me here.
Oh, wonderful. Next time on Lunch Break Science, we meet paleoanthropologist Dr. Bence Viola, who will be joining us from Austria and learn about the intersections of Neanderthals, Denisovans, and early modern humans. We'll see you there on August 18th. It'll be a really wonderful episode, so don't miss it. Thank you all for taking a break from your day and feeding your brain with the Leakey Foundation. Until next time, stay hungry for knowledge.
Bye-bye. Thank you.
Lunch Break Science is brought to you by the Leakey Foundation and is made possible by the generous support of the Anne and Gordon Getty Foundation, Camilla and George Smith, and the Joan and Arnold Travis Education Fund. Help raise $1,000 for the Baldwin Fellowship Program in celebration of our Executive Director, Cheryl Camisa Smith's birthday. Donations to the Baldwin Fellowship Program will be quadruple matched by generous sponsors, meaning your impact will be quadrupled. A $25 donation will have $100 worth of impact. Subscribe to the Leakey Foundation's YouTube channel, follow us on Facebook and Twitter, or sign up for our newsletter to be the first to hear about exciting upcoming programs and research. Miss an episode of Lunch Break Science? Catch up on past episodes and browse our library of Leakey Foundation lectures on our YouTube channel. Still hungry for science and can't wait till our next episode? Check out Origin Stories, the Leakey Foundation's award-winning podcast. If you enjoyed today's episode of Lunch Break Science, be sure to check out Origin Stories' first episode, On 2 Feet, featuring today's guest, Dr. Carol Ward. Origin Stories is available wherever you listen to podcasts. Visit us at leakeyfoundation.org to learn more about the Leakey Foundation, today's guest scientists, our educational programs, research grant and scholarship opportunities, human evolution news, how you can help support human evolution research and programs, and much, much more. Thank you for watching, and see you next time.